Welcome everyone to the fifth webinar organized by the ECIs. My name is Rafaela. I'm based in Portugal, Lisbon, at the National Institute of Health, Ricardo, Jorge. It's my pleasure to host this webinar here on behalf of the whole organizing committee. I will now present our speakers today. I'll just have to share my screen. So today we are going, our topic today is cancer molecular biology, which is a broad topic, but making the long story short, we will have an introduction by Lorenzo Montanaro. Lorenzo is a medical doctor and a full professor at the Department of Medical and Surgical Sciences at the University of Bologna in Italy. And he's also the director of the second cycle degree in medical biotechnology. And he's mostly interested in studying potential biomarkers with the purpose of using them as diagnostic tools. Then we will hear Federico Zacchini, who is a teaching tutor at the same department at the University of Bologna. Federico has a degree in biological sciences and a PhD in medical sciences. And he's going to talk to us about small non-coding RNAs and their role in retaining transcripts in cancer. Finally, Catarina Ramalho, a PhD student at the National Institute of Health, Dr. Ricardo Jorge, here in Lisbon as well, the same as I, will talk to us about the delta 160 p53 protein, a shorter p53 isoform, and their role, each role has an oncoprotein protein. So without further ado, Lorenzo, thank you for accepting to be here. The floor is yours.
Okay, thank you very much, Rafaela. I would like to thank the ECI Organizing Committee for giving the opportunity to give this introduction, and of course you for your nice presentation. Now I'm starting to share my screen. I hope it works fine. So I hope you are seeing the screen because I'm not seeing it. Okay.
Yeah, it's perfect
do you
now.
see my screen now?
Yes, perfect now.
Okay, thanks. So again, thanks for your nice introduction. I would like to, let's say, give a brief introduction on snoRNA and cancer. I know that also here they are experts in the field, so I mean, I'm very glad of their presence and maybe of their suggestion at the end of the presentation. And I would start with a very basic concept that is known by all the people studying, let's say, the protein synthetic machinery in cancer, that one of the features of neoplastic cells that have been recognized by pathologists more than a century ago is the prominency of the nucleolus. And now we know, also with a certain grade of information, that this prominency of the nucleolus is mostly due by the hyperactivation of the, sorry, I can go on. My screen, okay. This is due mainly to the hyperactivation of the process of ribosome production. So we know that cancer cells need, of course, in order to maintain the protein synthetic demand, to fulfill the protein synthetic demand, they need, of course, to upregulate and maybe also to modulate the process of ribosome production. And even this slide that is very, schematic or simple, resembling the different steps that are involved in ribosome production, there are at least two steps in which a group of non-coding RNA are involved. That is the modification of ribosomal RNA and its processing. In fact, more than 30 years ago, there's been the say, identified a group of small non-coding RNA that localized in the nucleolus that are involved in the processing. First, this is the first function has been identified, and then in the modification of ribosomal RNA. And then this small nucleolar RNA, so snoRNAs, has been characterized deeper and deeper, and now we divide them basically on the basis of their sequence into two major groups. So the so-called CD box snoRNAs that associate with a number of core proteins to form CD box small nuclear ribonucleoproteins. And these proteins include also the methyltransferase fibrillin, which is able to site-specific methylate ribosomal residues at 2 positions, mostly on ribosomal. And the other group of snoRNAs is represented by the HACA box snoRNA that associated with a distinct set of ribonuclear of core proteins, including the pseudouridine synthase discarin, that are able to mediate the site-specific isomerization of uridines into pseudouridines. Now, these two sets, of course, of snoRNAs and small nuclear RNPs are mostly localized in the nucleolus, so they take their name on their localization, but they are also localized. They can also localize in the cacao bodies. So for this reason, they are called snoRNAs or small cacao bodies RNAs. And as I said, for CD box, there is also a recognized role in the processing of ribosomal RNA from the premature, let's say, forms to the mature forms, and for both of the classes, they are, of course, involved in the modification process. Now, there is some additional information to add that concerns the gene organization of this RNA, because in humans, in mammals, and from vertebrates on, basically the genes which transcribed for the snoRNAs are hosted into coding genes, mostly coding genes that are coding for proteins involved in the protein synthesis or translational apparatus. So the snoRNAs are transcribed together with all the genes by RNA polymerase II. This occurs for most of the snoRNAs, for the large majority of the snoRNAs, and then they are processed by the removal of introns, and in one case, this process can lead to the formation of the mature mRNA encoding for one of the proteins, let's say, encoded by the host gene, and let's say in other case, this can be processed and trained and let's say generate the mature snoRNAs that then localize in the nucleolus and carries out the function that I just described. So according to this, it's not surprising also to know that the snoRNA sequence can be present in the cells not only in the form of the mature small nuclear RNAs, but also in the form of longer non-coding transcripts, which are represented by the transcripts from the host genes that retain the introns containing the snoRNA sequences. So these transcripts have been described since, let's say, many years, but recently function has been ascribed to them. For instance, we have the so-called snoRNA containing long non-coding RNAs. Among them, there is the so-called SLERT, that stands for snoRNA-ended long non-coding RNA enhancing pre-RNA transcription. These are all, for instance, the snoRNA-ended polyadenylated transcript that can be localized. Sorry, I try to activate the pointer here. Okay, that can again can be localized in the nucleolus, but also in addition to them, we also know that a considerable amount of transcripts retaining, or say isoforms of the host gene, retaining the snoRNA isoforms, are transcribed and then exported as mature but non-coding mRNA in the cytoplasm, where they are recruited to polysomes. They are initially translated and recognized as non-coding, and then degraded by the RNA decade mechanism, leading in some cases to shorter version of this snoRNA retaining transcripts, represented only by their 3' end. Okay. So coming to the more, let's say, actual point of snoRNA and cancer, this is just a very recent PubMed search with the keywords snoRNA and cancer. And as you see that, I mean, after the discovery, the topic remained in a niche, but after 2010, they became more and more popular. So at the end of the 2010s, we had hundreds of published reports concerning snoRNA and cancer. And then after the pandemic, I can see also a decrease in the number of publications. But I would say that the interest in this sequence has been represented mostly by the fact that specific association could be found between the expression of some snoRNAs and cancer features or prognostic features of the patients. Also, these snoRNAs are extremely stable, so it can be measured quantitatively, also in body fluids, for instance, or in tissue specimens, and associated with, as I said, many cancer-specific features. So they become, let's say, very popular, and they attracted an interest that a follower can be considered similar to what happened to microRNAs. However, we have to take this data with some caution, since there are many technical issues concerning the quantification and the use of this data in this sense, at least so far. First of all, there are, as I said, technical issues that are related to the size of these snoRNAs that are at the bridge between small RNAs and, let's say, longer RNAs. So the extraction issue is a problem. I mean, of course, it can be done, but not all data sets can be used because of the extraction system that has been used. That this could introduce a bias in that. And then also, since they are highly structured, also their reverse transcription, library preparation, or in other quantification approaches can lead to, let's say, quantification bias, also to flatten the difference in the expression of these specific transcripts. Also, we also find them in many transcriptomic data taking into account the poly-A. The poly-A transcripts. It is very surprising, considering that the features, of course, in the sequence of the canonical snoRNA, and is less surprising considering the existence also of longer polyadenylated transcripts. However, this has to be, say, considered when analyzing sequencing data, especially if there are data coming from the analysis of short sequences. And let's say more importantly, there is a really limited amount of studies compared to the hundreds that I just reported that gave functional insights into the changes of snoRNA expression in cancer. So it's very, let's say, there are many association studies, many studies linking the features of the tumors with snoRNA expression, but just a limited number of them that really provided functional insights into for their role. So, of course, there are studies providing information. So I'm not reporting all of them, but I'm just trying to make a summary of studies that really could demonstrate a role of snoRNA in cancer according to their function. So we know that the alteration in the function and in the expression of snoRNA can lead to alteration because of their guide function. For instance, on a ribosomal RNA, we know that the alteration in the expression of some snoRNA can lead to an altered, a decreased, mainly, or also an increased modification to in some specific RNA, ribosomal RNA residues, leading to translational changes. So basically changes in gene expression at the post-transcriptional level, especially at the translational level. But also we know that this alteration in the guide function could also involve different substrates, for instance, mRNAs. That is a very open and interesting topic this time. So snoRNA can guide the modification also at the mRNA level, also at the tRNA level, and it has been reported also for other non-coding RNA, including small non-coding RNAs. Small non-coding RNAs, for instance, lysosomal small nuclear RNAs. But we also know that snoRNA can interact with proteins. So of course they are, let's say, RNA molecules that are able to bind proteins and can interact with proteins, changing their function. The important reports on the interaction with the SNORD50 snoRNA with KRAS protein inhibiting its activation. So this is independent of the guide function, independent on the sequence-to-sequence interaction, but more dependent on the RNA protein binding affinity with the snoRNA sequence. But this is true for the canonical snoRNA, but this can be also considered true for the snoRNA retaining transcripts. So there are a limited number of studies, but in some studies this is also true for both CD-box and for transcript, both retaining CD-box and HAC box snoRNA that can both interact with a number of other mRNA. So presumably, even though this is not proven yet on the basis of their guide properties, but also interact with a number of proteins. So this can be, I think, a field for future studies in order also to investigate more their role in the cells. So I think I finished. Also, I trespassed my time, so I quit here, and I don't know, and I give you back the word to Rafaela. Thank you again for your attention.
For presentation, I think they're all much more elucidated about what the small nuclear RNAs are and how they contribute to cancer development now. I'll move to Federico. I think it's hello, Federico. Thank you for accepting to deliver a talk today. I think it'll be the logical sequence now to listen to your talk about the great introduction after the great introduction Lorenzo just did. So whenever you like, please share your screen.
Okay, thank you. Yeah, we'll continue the story that Lorenzo already start to introduce. So you should see my screen now, probably.
Yes.
Okay. So yeah, I would like to thank you for the opportunity to introduce my study, but this is basically I want to continue the story that Lorenzo introduced, but regarding more on the so-called snoRNA retaining transcript and their involvement in cancer development. So I don't want to spend time on what Lorenzo already told, but just to introduce again that starting from host gene, you can usually have say the canonical RNA for the host gene and the snoRNA sequence after the splicing process. But in some case, you can have a retention of the snoRNA in introns that ending up with this transcript that we call the snoRT, so snoRNA retaining transcripts. But how this transcript can be, let's say, related to cancer? We started with the basic idea that the modification of pseudouracilation complex could lead to a formation of dysfunctional ribosomes. This is true for example, dysregulation of discarin in breast cancer cell could lead at modification on the level of the pseudouracilation of the ribosome that can lead to different translations of some messenger RNA that could be related for cancer progression, for example. And this is already known, but an unbiased approach aimed to identify which messenger RNA are regulated after the modification of discarin, for example, that is the pseudouracilation synthase, of course, was never performed. So basically, we started by silencing discarin and doing polysomal fractioning in order to quantify by RNA-seq the level of the translated messenger RNAs. But what we basically found that most of these transcripts, many of these transcripts at least, have a significant feature in common. That is the fact that they retain the sequence of a snoRNA in some of their introns. So we have an enrichment of these snoRTs. And so because we know that discarin is related to cancer progression, the dysfunction of discarin is related to cancer progression, we thought that maybe could be also relevant these transcripts on the cancer progression. So the other things that is curious that since the polysomes are of course cytoplasmic, are present in cytoplasm, while snoRNA are usually located on the nucleolus. We wanted to understand better the context, so we did RNA immunoprecipitation of discarin after subcellular fractionator in order to see if we can find these snoRTs on the cytoplasmic portion of the cells. And what we obtained was that surprisingly, discarin is present also in the cytoplasm, even of course in a lower amount compared to nucleus. But after analyzing the RNA associated with discarin, we found that all of the snoR that we found with the RNA-seq was highly enriched, preferential in the cytoplasm. This is the presence of this node was not completely new, because in a few years ago, a research group published a work about the presence of these nodes regarding the SCD box SNORT of the NOP50C60C gene, in which they found that this SNORT is not only present, but also is further processed to a small version. This characteristic to have that's handing with the snoRNA sequence, and also this process can lead to a regulation of its own host gene. So we thought that maybe we are in some kind of the same in the same case, but regarding SCD box snoRNAs. So we further analyze our target, and by sequencing a 5' race, we aim to identify that basically all of the Hawa's nodes are actually present in two type of transcripts, in two type of isoform. One is the full length isoform, while the other is this short fragment that end basically with the sequence of the snoRNA. And we further characterize the distribution of these two isoforms by doing this, the PCR, we saw that the most abundant portion is this shorter fragment compared to the full length. And also this shorter fragment is also more enriched in the cytoplasm. It's basically the only present in the cytoplasm. So we thought also that maybe this transcript is, this shorter fragment is the most relevant of the two. But in order to better characterize these kind of isoforms, we also analyzed again polysomal fractioning on MCF7 cells, looking at 5 different kind of snoRs, 4 HAC box and also 1 CD box, derived snoRs. And what we obtained basically is that the full lengths are almost recruited to polysomes, so it's translated by ribosomes, while these shorter fragment, these 3' snoRs, called the 3' snoRs, are basically more abundant in the prepolysomal fraction. So this is further suggests us that they are actively translated, and then with some kind of RNA decay mechanism like non-transmitted decay or no-go decay, they can be further processed to a smaller version. This is also further validated, let's say, with another kind of experiment. We treated the cells with silvestrol, that is a specific inhibitor of initiation of the translation. And in this case, you can see that the full length level of the snoRs, of these 4 different snoRs, increase after the treatment, meaning that if they are not recruited to ribosomes and translated, they cannot be more processed, so they accumulate. This is not only suggests that they are actively translated, but also that, of course, this kind of RNA species is way different from what is known regarding snoRNA. So we are talking about different kind of transcripts that can maybe related to cancer. So we will further show you the data about these things. I want to put attention on one thing. These kind of transcripts are not, why these kind of transcripts are not described much in literature. The major problem is that, for the reason that also Lorenzo told you earlier, is that these transcripts are not easily detectable with the standard massive RNA sequencing. So by taking advantage of nanopore technology, you can discriminate that in which you can have the full transcript read. You can easily distinguish with both between the 2 type of transcripts. So we analyze a data set of Oxford nanopore sequencing for 5 different cell types. And thanks to this analysis, we analyze basically all the annotated snoRNAs that are more than 600. And we found that many of these have are retaining some intron in some way as only full length or only short fragment or in both form. And of course, this distribution is a bit different between the cell lines, but it's still present in all of these kind of tumor-derived cell lines. But by looking specifically to the K562 cell line, we also have the data, analyze the data of the subcellular fractioning on nucleus and cytoplasm, and we also here have this distribution of the snoRt fragments both in nucleus but also in the cytoplasm. So this is further evidence that these snoRs are widely distributed among the cell lines. So I told that I said that this screen is known to be related to the cancer progression. We know that this screen regulates SNOT some way, and this screen also interact with SNOT with the first immunoprecipitation that I showed you. So to have a more complete picture of what can be bound to this screen, we did immunoprecipitation sequencing of cytoplasm in this screen, so specifically the cytoplasmic version of the subcellular fractioning. And we found that more than 700 transcripts are attached to this screen, in which 50 of them are basically SNORT, but the majority, and they are among the most abundant one, but the majority are related to are protein coding, but they are among the lower enriched, less enriched. So why we have this high percentage of protein coding? Actually, we don't have the answer, but we have a suggestion that maybe these SNORTs can interact with the pseudoregulation complex and could guide, could interact with messenger RNA and potentially also guiding the modification of messenger RNA, because it's known that messenger RNA can be pseudoregulated some way, and impacting on the expression, regulation, or stability of messenger RNA. Because the messenger RNAs could be the coding messenger RNA could be the interested subtype, we did a gene ontology analysis specifically on the protein coding transcripts, finding basically an enrichment on these two categories that are related to hormone receptor functions both. And because we are talking about the study on MCL7 cell, the estrogen-dependent cell line derived from estrogen-dependent tumors, we then decide to see if they are regulated in some way these transcripts after this screen silencing. So we then silenced it. We did an acute depletion of this screen, and we tested we look at the expression of this transcript, and we found that most of these are some way regulated by the silencing of this screen. So so maybe this could be a way of the cells to have some regulation of transcript that is related to estrogen dependency. And in fact, analyzing these regulated transcripts on a public data set, on the CGA data set of breast cancer patients, we found that a correlation with the survival of a specific subtype of patient, this luminal B, that have the characteristic to be estrogen and progesterone positive, like also the luminal A, but also usually have a worse prognosis. So again, we think that maybe this so-called cytoplasmic discrete function, let's say, that can be related to the interaction with SNOT, can be affect the estrogen dependency of breast cancer. So because we know that one of these transcripts, that is the EA41 SNOT, is the most regulated after the screen depletion, and this is also the most abundant. We thought to use it as, let's say, a proxy of this cytoplasmic activity of the screen. So we tested this expression on a series of patients, breast cancer patient, and we found a strong correlation with the survival, specifically with the estrogen and progesterone positive status, receptor status patients, further suggesting that could be some dependency with the hormone dependency of this patient and the expression of these SNORTs. To further go with this idea, and having an evidence in vivo, we also did a xenograft of MCF7 cells in nude mice without the estrogen treatment. MCS7 are known to be tumorigenic, but only upon a continuous estrogen treatment. So why not giving estrogen treatment, you expect not having many tumorigenic potential. And as you can see here, there's just a few the tumors that was developed after the subcutaneous injection, while if by the injection with the discrete silenced cells, these the tumorigenic potential is way more higher. So again, this is suggests that the regulation of this screen can be related to some, let's say, estrogen dependence growth of these tumors. So now that we have some evidences of the relation between these nodes, at least a few of these nodes and cancer, we try to find a way to regulate them. So basically, because they are cytoplasmic, and of course they are derived from messenger RNA, we try to knock down using siRNAs approach. Specifically on breast cancer cell line, in this case, we had two cell lines that are positive for estrogen and progesterone receptor, while one, the MB231, that is a triple negative. So this is a negative also for both the hormone receptors. And why we achieve a good amount of silencing on all of these tested snoRT, these don't affect the full length expression of these snoRT, even if they share in the same sequence. So these full lengths are targeted by siRNAs. Probably this result is because most of the isoform that is present in the cell line is the shorter version. So this is also probably the shorter version is probably the most affected also by the silencing. There's another curious thing that we also tested the host gene, the coding transcript, basically the host gene of these snoRTs. So these coding transcripts don't share any target sequence by these siRNAs, but still they are a bit downregulated. And this is in line in which what shown by some other authors for the CD box snoRTs. But this is also curious because the majority of the snoRT host gene are belonging to genes that are highly important for translation, generally speaking, because we are talking about, for example, of translation initiation factor or ribosomal protein. So regulation of snoRT, if it can impact also on the regulation on the coding transcript, this could be, of course, relevant for the studies. So after achieving this good silencing, we simply test some tumorigenic features, so the cell growth and echronogenic development. And basically what we found that after the silencing of these transcripts, there's some changes on the growth cell, on the growth of the cells and the echronogenic potential, but only in estrogen and progesterone positive cell line. So again, there's this correlation between the snoRT expression and the tumorigenic potential, specifically on this kind of cell lines. So just want to conclude my brief talk. I show you that these transcripts that retain the sequence of snoRNA are present in the cytoplasm, bind to this screen, and are also regulated by this screen. And not only is present on the cytoplasm, but are actually translated and then processed to a shortened fragment. The common mechanism is not already known. For some of these, probably is the non-seismic decay mechanism, but could be also other kind of mechanism like no-go decay, for example. We also know that the regulation of this node can affect the expression of the host gene. and can impact in some cellular tumorigenic features like clonogenic or cell growth rate. And I show you also that the regulation on these or some messenger RNA that could be mediated by this snoR transcript could be relevant for the nuclear hormone receptor function and also can influence the behavior of breast cancer cells. So just to finish, I want to thank you, of course, my colleagues, the Lorenzo Montanaro groups, the Mariano Pencil group, and the collaborator of the CB of Trento, and of course the funding partner as ICAN Palotti Legacy. So thank you for your attention, and yeah, I think that I finished my talk.
Thank you so much, Federico. Great talk. So does anyone have questions for Federico? Questions? We have time for a couple of questions.
I can ask a question. I don't know if you hear me.
Yeah.
Yes.
Nice presentation.
Thank you.
A quick question about the experiment where you did the depletion of DKC1.
Yes.
And you depleted DKC1, and then you assessed the expression of different transcripts which belong to the nuclear receptor coactivator and nuclear hormone receptor binding families.
Yeah.
And my question is about if you check ribosome biogenesis in these DKC1 depleted cells. Sub-question being, for how long did you deplete DKC1 in these cells?
Okay, so I can start with the second question. We standardize our depletion for 72 hours. That we found that is the time where the D-screen is the most dysregulated, unregulated in this case. We know that probably this time is not enough for having a renewal of ribosomes. So after 72 hours, the ribosomes cannot be probably much affected by the pseudoregulation. At least they are not completely changed. So the silencing on the screen should not be so relevant, probably on the general pseudoregulation level of these ribosomes. But we didn't check the ribosome yeah. So you suggest that could be
will be a good control, I think, to do. Yeah. And I have another quick question about the about whether you think that the snoRs, the cytoplasmic snoRs, do you think that they assemble all 4 core proteins onto the box, in your case, HACA motif?
Yeah.
Because these motifs and the proteins that bind to them are very well known to target the RNAs that display them to the nucleolus. So it's a very strong nucleolar targeting signal. And so the question is to know, there's a question about how they remain in the cytoplasm. I mean, first of all, how they go to the cytoplasm and how they remain there, because the box CD or the box HACA, once bound to the core proteins, this is a super strong targeting signal to the nucleolus. And so I was just wondering if you know if all 4 core proteins actually assemble on these motifs, or you mean DKC1, but I don't know about the other ones.
Yeah, this question we can answer because we did the RNA monoprecipitation also for the other core protein, looking only of some targets. So we didn't do the RNA, complete RNA-seq, but we tested some snoRs also for the other core protein, and we found on the cytoplasmic, after cytoplasmic fractioning, and we found the same result basically. And we also separately tested also the we purify ribosomes, generally speaking, and looking for also the proteins. These also all the proteins of the co-protein of pseudoregulation complex are, let's say, found also purified with ribosome using not high salt purification, so with a low salt purification that can have, of course, also the other initiation factor or translational relevant proteins. So we think that we have different suggestions that say that the core protein are all binded to the snoRs. Why they are outside is not clear at all at this moment.
Thank you so much. Very interesting.
Thank you.
Thank you. Okay, Elena, please.
Hi, a very interesting presentation. Thank you. I had a question about your results from the nanopore sequencing. If I understood correctly, so you're saying that there is much more of this snoRNA transcripts present in the cell than what we see by traditional sequencing. But I was just wondering how can you discriminate those from nascent transcripts that are present in the cell? Because we know from data, the sequencing data that has been generated by using random priming, that then all of a sudden you can really see a lot of nascent transcript present in the cell as well. So to what extent is what you see in the Oxford Nanopore nascent transcript versus actual real snoRNAs?
Yeah, we, in order to discriminate from a nascent transcript, we basically look at the reads of these full length of these these complete reads, and basically we discriminate the nascent the snoRNA only when they are present that single introns is retained, not all the introns. And to discriminate between the shorter version, we simply checked for the reads that don't have these the 5' parts of the transcript, but only transcript that start with the snoRNA sequence and go along with the end of the transcript, basically.
Okay, thank you.
I have
Any more questions?
May I have a question? Hi.
Yes, of course, Paulina.
Have you considered looking at the actually chromatin-associated nascent RNAs? Because a lot of splicing is happening co-transcriptionally. So actually, this is the tool where you can actually see whether splicing of snoRNAs is happening and this isoform when it is actually generated, whether it's delayed or it's like happening within, the window of co-transcriptional splicing.
Yeah, this is a good suggestion. We never checked on this on the association of chromatin, but we know that, for example, some of these nuclear snoRNA, let's say, are co-transcriptionally originated, because there's other research group demonstrate these things, but they demonstrate for a nuclear transcript that these nodes stay in the nucleus. So this could be a possibility, of course, and probably worth checking.
I have a question.
Thank you. Go ahead, please.
Thank you. It was really nice presentation. A question, just a technical question, because we have recently started working with snoRNAs, CD-box specifically, and we were trying to amplify the snoRNA expression using different extraction method for RNA, because what we what we understood that the enrichment of these snoRNAs can be different based on the extraction technology, but we couldn't conclude actually that which extraction technique can be the best one. So we tried for trisol extraction as well as small RNA kit that we get from QIAGEN, and the others one is for full length RNA extraction from QIAGEN as well. So we do see expressions in all the three extraction methods, but I think what we saw is that in small RNA extraction method was the enrichment was better, but it was not far better. I mean, it was not that better that we could conclude that which one is the best one. So can you suggest some?
Yeah, regarding the best method, I don't have much suggestion, because we found something similar. So we struggle also to have a good extraction method for snoRNA. We focalized more on the snoR that are longer transcripts. So for that case, I think the RNA extraction is less impacted by using, let's say, trisol. compared to the standard snoRNA. But I want to just add one other thing that we found that probably more than the extraction is also the reverse transcription of the RNA. So the snoRNA that can be really different if depending on the type of reverse transcriptase that you're using. So we usually suggest using a reverse transcriptase that goes to higher temperature in order to have less chance to be affected by the secondary structure as snoRNA. But yeah, related to the purification, we don't have much suggestion, let's say.
Okay, and the second question is, I saw that your host gene expression was also affected. We also got the same, and we were thinking that maybe the knockdowns were not very specific, because of that the host gene expression was also being affected. But as you said, it could be different explanation for it. So I was wondering.
Yeah, actually, because these knots, depending on the type of knots that we are talking about, could be abundant. So if you are using some kind of, let's say, an ASO to silence the snoRNA, you can actually end up silencing also a bit these knots that at least for some of these can affect some way the host gene. We need to demonstrate how it works, but at least for a CD box snoRNA, this NOP56 derived gene, they are demonstrated that there's this feedback loop that affect also the host gene, specifically silencing the cytoplasmic snort. So if you are using a general mechanism of silencing, maybe you are impacting not only the snoRNA, but also the snort. Okay.
And the third question, which is the last one, regarding the knockdown technology that you have used siRNA to knock down these snoRNAs.
Yes.
So did you just came up with the siRNA technology, or you also tested others? Because in literature, there are several techniques that can be used, and it can be different outcomes. So we started using antisense oligonucleotides for these snoRNAs. And we were also thinking that maybe we could have taken checked the siRNAs as well, but there are many studies that they show that the knockdown is not that efficient. So what is your experience in that?
We use SNAs because we want to silence specifically the cytoplasmic transcript, not the nuclear one. So by using SNA, you are more sure that you are not affecting the nuclear parts. Why, if you want to go, of course, to silencing snoRNA, you need to use ASO. You don't have any other option, I think. And it's difficult to silencing, but we also achieve a way for the study to silence also snoRNA, but is really dependent on the type of ASO that you are testing.
Okay, thank you.
Thank you. Thank you. We are running short on time, so I suggest we move. Thank you, Federico. So I suggest we move to Catarina now. So Catarina, please share your screen and whenever you like.
Hi. Can you see the full screen now?
Yes, please.
Okay.
Go ahead.
So hi everyone. I want to thank the organization and Rafaela for inviting me to give this presentation. So my name is Catarina, and I'm a PhD student at two groups. I'm doing work in these two groups: mRNA metabolism and associated diseases group, headed by Luisa Romão in Lisbon, and then my main supervisor is Marco Candeias, who is head of the Molecular and RNA Cancer Unit at the University of Kyoto. And so today I'll be presenting the work from Marco's group and from that I've been developing during my PhD, and also the work from different elements from the group, including Rafaela, actually. And it is about delta 160p53, which is a short isoform of p53. So I'll start with a brief introduction about p53. The p53 protein is a transcription factor. It can activate different tumor suppressor genes in response to different types of cellular stresses, such as DNA damage, nutrient starvation, or oncogenic signaling. And it forms tetramers on the response elements in the promoters of its target genes. And then among its many different functions in the cell, the first ones to be described and the best characterized are regarding the cell cycle arrest, the promotion of cell cycle arrest, senescence, and apoptosis. And because of that, it is commonly called the guardian of the genome, because the activation of p53 when there is DNA damage is very important to promote the repair or the elimination of the cell, and in this way avoiding the propagation of potentially tumorigenic cells. However, it is also a proto-oncogene because the p53 gene is found mutated in more than half of all human cancers. And in most of the cases, the alterations that are observed correspond to missense mutations, which is not very common in tumor suppressor genes. And these mutations can lead to the loss of the wild type tumor suppressor functions. So this is lost in the cell. Or it can actually lead to the gain of new functions that will actively drive tumorigenesis, invasion, and metastasis. And in this case, these are called gain of function mutants. But the p53 gene does not encode only for one protein, but actually for at least 12 protein isoforms that result from transcription from 2 different promoters, from alternative splicing, or even from internal initiation of translation. So the functions that I mentioned in the previous slide were mostly attributed to full-length p53 alpha. So then, due to alternative translation, alternative splicing, there can also be 2 other C-terminal variants of each protein. So we have the alpha, beta, and gamma variants, and the most expressed ones are typically the alpha ones. And then we also have shorter isoforms that collect some terminal portion of the full-length protein when compared to the full-length protein, and these are delta 40, delta 133, and delta 160. And these isoforms have also been extensively studied, and they've been shown to modulate the tumor suppressor functions of the full-length protein, and they do so in a tissue-specific manner. And actually, the shorter isoforms, delta 160 and delta 133, and also delta 133 beta, have been shown to promote proliferation, invasion, and metastasis. And so we see that there are already oncogenic roles present in the wild type p53, and so we think it is wrong to say that the mutants should be called gain of function. Now, about delta 160 in particular. This isoform is overexpressed in cancer cells, and it presents many of the oncogenic roles that are usually attributed to the gain of function p53 mutants. And it is, if we think it can be considered a proto-oncogene because the missense mutations that are usually found in the tumor cells actually promote oncogenesis by favoring the translation of this isoform. And this will actually provide an explanation of evolutionary origin for the oncogenic functions that are found within p53. And we actually have observed that the start codon for delta 160 is very well conserved in mammals. Here we have species from different orders within the mammal class. And you can see that the start codon for delta 133 is actually only conserved in primates. And there's still a lot that is not known about delta 160. It's also what factors its translation and when and how it is translated. And we have found that the integrated stress response can actually promote the expression of Delta 160. So just to give some context about this program, the ISR is mainly a pro-survival program that is activated in response to different kinds of stresses. Here in our case, we are working mostly with ER stress, and then different specialized kinases are activated in response to these different stresses, and they will lead to the phosphorylation of EIF2 alpha. And so when that happens, EIF2 alpha, when it is phosphorylated, it cannot participate in the formation of the ternary complex, which is necessary for the canonical cup-dependent translation. And so what happens in this situation is that during ISR is that there is a global reduction of protein synthesis, as this is blocked. And but actually there is going to be alternative translation of some mRNAs. And so one of the ISR effector, one of the first mRNAs to be, or the proteins to be activated during the ISR is ATF4. It is also a transcription factor, and it will then promote the translation of other, the transcription of other genes, so that they are also